Hello and welcome to Cell Replication and Cell Division by Drake, Margot, Hannah, and Harry. The first thing we'll be talking about is why cells replicate. Well, cells in our body are constantly being replaced and created. Now you might be wondering, how is this done? Well, there are two biological processes by which new cells are made. The first is mitosis. Mitosis is the process by which one cell replicates by dividing into two identical cells, or sister cells, identical to the cell they stem from. Mitosis enables growth and also allows damaged cells to be replaced, with the cell itself finally splitting into two. Here we are modeling mitosis in an organism with two pairs of chromosomes, one which comes from the father and one which comes from the mother. Reminder, humans have 23 pairs of chromosomes. The second is meiosis. Meiosis is the other process by which cells replicate, and it is responsible for creating gametes, or sex cells. Unlike mitosis, which makes two cells with both copies of all chromosomes called diploid cells, meiosis results in cells with one copy of each chromosome called haploid cells. Meiosis is necessary because it ensures that all organisms produced through sexual reproduction contain the correct number of chromosomes. Since two gametes, an egg and a sperm, come together to make one embryo, each gamete for this organism only has 3 chromosomes. But before meiosis officially begins, DNA must replicate. This occurs during a stage called interphase, when the cell grows and prepares for division. The most important part of interphase is DNA replication. Meiosis can be divided into two main events, meiosis 1 and meiosis 2. Pretty simple, and further broken down into 8 total events. The first phase of meiosis 1 is prophase 1. Chromosomes which are in pairs with their sister chromatids condense into an X shape and then find their homologous pair through synapsis, which fuses the chromosome pairs together at the start of meiosis to form them into tetrads, meaning a group of 4. It is during prophase 1 that homologous recombination occurs, when genetic material gets exchanged between homologous pairs. There will be more detail on this later, so get ready. During prophase 1, the nuclear membrane that confines all of the DNA dissolves so it can move freely through the cell. The next phase is metaphase 1, where homologous pairs line up at the center of the cell. The orientation, which allows a cell to get some of chromosomes from one parent and some from another. At this stage of meiosis, the meiotic spindle fibers are fully formed. Spindle fibers, which extend from centrioles, attach to the chromosomes. After metaphase 1, anaphase 1 is brought into action. This is where homologous pairs begin to be pulled apart to opposite sides of the cell by the spindle fibers. However, the sister chromatids are still together. The final phase of meiosis 1 is telophase 1. Homologous chromosomes reach opposite ends of the cell, and a new nuclear envelope forms at the end containing 23 pairs of chromosomes. We will be modeling meiosis 2 as one part instead of doing 2 daughter cells because they are both completing the same task, so we figured why not just analyze one, that way it's more clear, but we will show what it looks like in the end, of course. Meiosis 2 is performed in a similar manner to meiosis 1. In prophase 2, metaphase 2, anaphase 2, and telophase 2 in cytokinesis. In prophase 2, we have our 2 daughter cells, each consisted of 23 chromatid pairs. The membrane around the nucleus in each daughter cell dissolves away, releasing the chromosomes. In metaphase 2, the daughter cells line up in their equator of the cell, and the meiotic spindle fibers at each pole of the cell attach to each of the sister chromatids. In anaphase 2, which is where the chromatids become individual chromosomes. This happens by the sister chromatids being pulled to opposite poles. Now it's time for telophase 2. The chromosomes complete their move to the opposite poles of the cell. A membrane forms around each set of chromosomes to create 2 new nuclei. Division of the cytoplasm during cytokinesis results in 4 haploid cells. Finally, cytokinesis is performed again. Fertilization occurs when a sperm and an egg combine and their nuclei fuse. Because of each of these reproductive cells is a haploid cell containing half of the genetic material needed to form a human being, their combination forms a diploid. The product of fertilization is a cell called a zygote, as you can see in this diagram here. Another thing to know is Mendelian genetics. Can we know what the offspring of 2 organisms will be like? Well, Gregor Mendel studied genetics using pea plants, whose traits can be traced to specific genes. For example, the color of the flowers in these plants is based on a single gene. Mendel studied the probability of getting a certain color flower based on the parent's flower colors. So each parent starts with 2 copies of each chromosome containing the gene that controls flower color. They're either homozygous, meaning both of their genes are dominant or both recessive, or they may be heterozygous, meaning they have one dominant gene and one recessive gene. Homozygous recessive flowers, modeled as small p, will be white. Homozygous dominant flowers, capital P, will be purple. And heterozygous flowers, capital P, will be purple. The presence of the dominant gene automatically means the flower will be purple. So we can't know the genotype of a purple flower just by looking at its phenotype, the expression of the gene. So in meiosis, the gamete produced by a parent gets one of their 2 chromosomes. For homozygous plants, it isn't a game of chance as to what kind of gene the gamete will get. Both genes are either dominant or recessive, meaning the gamete will either have one dominant gene if the parent is homozygous dominant or one recessive gene if the parent is homozygous recessive. However, for heterozygous parents, the gamete has a 50-50 chance of getting either a dominant or recessive gene. What's it going to be? The combination of the genes in these 2 gametes determines the flower's color. We can model this with a Punnett square right here, which helps us predict the probability of certain traits in offspring when the genotype of the parent are known. Here's the model. We mentioned earlier that during prophase 1, when chromosomes find their homologous pairs and fuse through synapses, the crossing over of genetic material can occur in a process known as homologous recombination. This means that one single chromosome in the final haploid cell could contain segments from both sets of parents. Recombination occurs when a holiday junction forms. A holiday junction is a cross-shaped structure that forms when segments of both homologous chromosome double strands are separated, resulting in a region with 4 single strands of DNA. So what we are modeling here is the molecular crossing over of homologous recombination, which, yes, of course, includes holiday junction. To begin, homologous chromosomes align. Then one bold step happens where a specialized protein makes a double-stranded break in one chromosome. After this occurs, the 5'3" nucleus digest one from each end of the cut, one nucleotide at a time. Next, the homologous DNA sequence is unzipped, which is followed by the strand invasion. Once that happens, DNA polymerase uses homologous chromosomes as the template to fill in the gap, the gray nucleotides. Then single-stranded cuts in the chromosome that was used as the template, red chromosomes. And finally, ligase joins and breaks in the DNA to form 2 recombinant chromosomes. And here is homologous recombination on a microscopic level. To conclude or summarize, cell replication is a vital process that ensures organisms can survive and reproduce. Through mitosis, organisms are able to grow and repair themselves. And through meiosis, cells can create little baby gametes. We also took a trip through Mendelian genetics, where we can try to predict what the offspring of 2 parents would be like. But remember, because of homologous recombination, offspring might end up being a total and complete mixture. Wow, genetics are crazy, man. Well, I hope you enjoyed this video just as much as we enjoyed demonstrating it for you. So much fun. Thank you for watching. Sites are below.